Hello friends, today in this video I am going to discuss about the MCQs on immunology, and this is the ninth part of the series of more than 1800 MCQs on immunology with detailed explanation. And this video will be beneficial for many life science exams like CSIR NET or ICMR NET or GATE Life Science or many MSc entrance exams. Okay, so let's start the MCQ session with detailed explanation on immunology. So the first, and this is the Telegram channel where you can follow me. Okay, so the first question is: a secondary humoral response is characterized by longer lag phase, no negative phase, longer persistence of IgG synthesis, or slow rise in antibody titer. And the right one is option C, that is longer persistence of IgG synthesis. So a secondary humoral response is characterized by longer persistence of IgG synthesis. So here we can see that when our body first exposed to an antigen, first time the IgM will appear. Okay, so when the same antigen expose when our body get the exposure of the same antigen second time, that time in second time the secondary humoral response, that is the IgG, came fast or appeared fast. Okay, and this during the secondary immune response the immune system can eliminate the antigen which has been encountered by the individual during the primary invasion, so more rapidly and efficiently. So in second time the immune system work more rapidly and efficiently. Both T and B memory cells contribute to the secondary response. Okay, so this is all about secondary humoral response in immunology. So the next question is: example of type 3 hypersensitivity is anaphylaxis, serum sickness, atopy, or contact dermatitis, and the right one is option B, that is serum sickness. So type 3 hypersensitivity occur when there is an excess of antigen leading to small immune complexes being formed that fix, complement, and are not cleared from the circulation. And this type 3 or immune complex hypersensitivity reaction are represented by tissue damage caused by the activation of complement in response to antigen antibody complexes that are deposited in tissues. Okay, so the next question is: Paul Burnell test is used for diagnosis of CMV inclusion disease, rubella, or infectious mononucleosis or measles. And the right answer about the Paul Burnell test is option C, that is infectious mononucleosis. So Paul Burnell test is used for the diagnosis of infectious mononucleosis, and the Paul Burnell test is used to screen for infectious mononucleosis, which is seen most commonly in adolescent and young adults, and this disease is caused by the Epstein-Barr virus. This infects B lymphocytes which subsequently produce a heterophile antibody which is identified through its reaction with the Paul Burnell antigen and is detected by a latex agglutination method. Okay, so move on to the next question, that is all the following are live vaccines except BCG, measles, Salk, or Typhoral. And the right answer is option C, that is Salk. So Salk is not a live vaccines. So a live virus vaccine contain a weakened live virus that is designed to help your body develop an immune response without you developing symptoms of the disease it is intended to prevent. So the virus is teaching your immune system what the virus looks like and allow the body to develop an immune system. So this is the live attenuated vaccine. Okay, so that is the live vaccine, so where the main virus is the live virus is injected, but the live virus is weakened, is formed, which is present in their weakened form. Okay, so they cannot produce that disease. Okay, so the Next question is, an example of neutralization test is Wasserman, VDRL or Paul Burnell or ASLO, and the right answer about the neutralization test is option D, ASLO. The term ASLO is an abbreviation for anti-streptolysin O antibody test, and it is a blood test carried out to detect the presence of antibodies in blood when the body is exposed to a bacterium known as group A streptococcus. Okay, so the next question is, proton phenomenon is due to antigen excess or antibody excess or proportionate level of antigen and antibody or no relation to levels of antigen and antibody. So the proton phenomenon is due to option B, that is antibody excess. So where the antibody is excess, so here a negative, so the proton phenomenon is a false negative response resulting from high antibody titer which interfere with formation of antigen antibody lattice. So necessary, which is necessary to visualize a positive flocculation test. So here if the antigen, if the antibody is excess, that time, so no lattice structure will form in antigen antibody flocculation test. So that is the negative, for false negative response due to the Proson's phenomenon where antibody is excess. Okay, so move on to the next question, that is HLA complex is located on the short arm of chromosome 6, chromosome 8, chromosome 2 or chromosome 5, and the right answer is option A, that is chromosome 6. So HLA complex is located on the short arm of chromosome 6, and this HLA means the human leukocyte antigen system or complex is a complex of genes on chromosome 6 in humans which encode cell surface protein responsible for the regulation of the immune system. So it is basically act as the histocompatibility complex or MHC, major histocompatibility complex or MHC molecule, which is mainly present in animals. So it is a like structure or a like system in human body. So HLA system is also known as the human version of the major histocompatibility complex found in many animals. Okay, so which reject the graft rejection, which is help in graft rejection. So or more specifically we can say it help us to detect which one is our self cell or which is our foreign cell, which is a foreign cell, which is our own cell. So to detect the correct one, so that is the that type of system. Okay, so move on to the next question, that is one of the direct activators of C3 component of complement is C1 esterase, C1 esterase and a cofactor or cobra venom and a cofactor or cobra venom, and the right one is option D, that is cobra venom. So one of the direct activator of C3 component of complement is cobra venom. Okay, so move on to the next question, that is when antibody and a soluble antigen combines, the reaction is agglutination, adsorption, hemagglutination or precipitation, and the right answer is option D, that is precipitation. So when antibody and a soluble antigen combine, that type of reaction is known as precipitation. So what is the difference between precipitation and agglutination? So the similarity is both are antibody cross-link, and both are the process where antibody and antigen complexes are present and precipitate done, but the difference is nature of antigen. So in precipitation soluble antigens are involved, and in agglutination insoluble antigens like RBC bacteria antigen fixed on beads. So this type of in agglutination insoluble antigens are present, or in precipitation soluble antigens are present. Okay, so the next question is, the routine serological test used for diagnosing syphilis is Wassermann or Kern or VDRL or TPHA test. And the right answer is option C, that is VDRL. So the routine serological test used for diagnosing syphilis is VDRL test. So that is the venereal disease research laboratory tests are slight microflocculation tests that detect antibodies produced against antigens released by damaged host cells in patients suffering from syphilis. Okay. So the next question is, passive immunity is durable and effective, has immunological memory, has a negative phase, or is transferred vertically, and the right answer is option D, that is passive immunity is transferred vertically. So passive immunization or passive immunotherapy is a process in which individuals receive antibodies from another source rather than producing those antibodies on their own. So passive immunity provides short-term protection against infection. Okay. Move on to the next question, that is, all are true about interleukin 1 except is a 15 to 17 kilodalton protein produced mainly by macrophages or binds to high affinity receptor on T cells by antigen MHC, or its receptor activates CD8 T cells, or its internalization readily induces RNA and protein synthesis of the responsive cell, and the right answer is option C, that is its receptor activates CD8 T cells. So all are true about interleukin except its receptor activates CD8 T cells. Okay, so interleukin 1 is a general name for two distinct proteins, interleukin 1 alpha and interleukin 1 beta, that are considered the first of a small but possibly growing family of regulatory and inflammatory cytokines. Okay, so the next question is, Russell bodies are present in macrophage, B cell, T cell, or plasma cell, and the right answer is option D, that is plasma cells. So Russell bodies are present in plasma cells, and these Russell bodies are inclusion bodies as usually found in atypical plasma cells that become known as Mott cells. Russell bodies are eosinophilic, homogeneous immunoglobulin containing inclusions as usually found in cells undergoing excessive synthesis of immunoglobulin and are found in the peripheral areas of tumors. Okay. Now move on to the next, that is HLA B27 is associated with ankylosing spondylitis or juvenile rheumatoid arthritis or coeliac disease or Reiter syndromes, and the right answer is option A. So HLA-B27 is associated with ankylosing spondylitis. Okay, and this human leukocyte antigen, that is HLA-B27, is a protein located on the surface of your white blood cells, and HLA-B27 is strongly associated with ankylosing spondylitis and other associated inflammatory diseases referred to as spondyloarthropathies. Okay, so move on to the next question, that is, plasma cells are not present in germinal center of lymph nodes, kidney, spleen, or lymphoid tissue of alimentary tract, and the right answer is option B, that is kidney. So plasma cells are not present in kidney. Okay, and this plasma cell are a type of white blood cell found within the bone marrow. Normally plasma cells make up about 5% of all cells in the bone marrow. They are part of the immune system, playing an important role in combating infection by producing disease fighting proteins called immunoglobulins or antibodies. Okay, so the next question is, the protein which binds to cell membrane in presence of calcium ion and create transmembrane pores, ultimately leading to death of the target cell, is Benz-Jones protein or alpha fetoprotein or porine protein or perforin, and the right answer is option D, that is perforin. So the protein which binds to cell membrane in presence of calcium ion and create transmembrane pores, ultimately leading to cell death, is perforin protein. So here this perforin protein binds with the calcium and form a active perforin form and are attached to the transmembrane and create a pore into the transmembrane and ultimately lead to the death of the cell. Okay. So the next question is coagglutination test uses Potak 1 strain, Cowan 1 strain, Nichols strain or Park Williams 8 strain, and the right answer is option B, that is Cowan 1 strain. So coagglutination test uses Cowan 1 strain. So what is co-agglutination test? So this is one type of agglutination test in which inert particles like latex bead or heat killed as Staphylococcus aureus or Cowan 1 strain with protein A, that type of inert particles are coated with antibody to any of a variety of antigens and then used to detect the antigen in specimens or in isolated bacteria. So, okay, so this is a one type of agglutination test. Okay, so move on to the next, that is ALEC test is a complement fixation test or a precipitation test or a neutralization test or an opsonization test. So ALEC test is option B, that is a precipitation test. So ALEC test and where an antigen antibody reaction in which the antigen is soluble. So this is a precipitation test. So here the antitoxin is present and here the bacterial growth is present. So bacteria, that is a toxin and antitoxin, diffuse out and they produce a precipitation at a certain line. Okay, so this is the ALEC test. So the next one is Nagler reaction is a toxin antitoxin neutralization test or complement fixation test or precipitation in gel or passive agglutination test, and the right one is option A, that is toxin antitoxin neutralization test. Okay, that is the Nagler reaction. So lecithinase test or Nagler reaction, which which identify organisms which liberate phospholipases, that is lecithinase, example Clostridium perfringens, and the alpha toxin of Clostridium perfringens has phospholipase activity and hence help in differentiation of Clostridium perfringens from other Clostridium species that also produce lecithinase, okay, by neutralization of lecithin C activity by an antitoxin. So move on to the next, that is Rose-Waller test is a precipitation in gel, reverse passive agglutination test, hemagglutination test or co-agglutination test, and the right one is option C, that is hemagglutination test. So Rose-Waller test is a hemagglutination test and this test is used for the diagnosis of an autoimmune disease termed as rheumatoid arthritis. The test is named after Dr. Eric Waller and Dr. H.M. Rose. So next question is COMTEST is a ring precipitation test or agglutination test or heterophile agglutination test or immunodiffusion test, and the right one is option B, that is agglutination test. So COMTEST is a agglutination test. So a COMTEST, also known as antiglobulin test, is either of two blood tests used in immunohematology, and they are the direct and indirect COM test. The direct COM test detects antibodies that are stuck to the surface of the red blood cells. Since these antibodies sometimes destroy red blood cells, a person can be anemic and this test can be helped clarify the condition. The indirect COM test antibodies that are floating freely in the blood. Okay, so this is the COM test. So next question is count test is a ring flocculation test, tube flocculation test, slide agglutination test or tube agglutination test, and the right one is option B, that is count test is a tube flocculation test. So count test for used for syphilis disease, a tube flocculation test. Okay, so move on to the next one, that is radial immunodiffusion is single diffusion in one dimension, double diffusion in one dimension, single diffusion in two dimension or double diffusion in two dimensions. So radial immunodiffusion is option C, that is a single diffusion in two dimension. So radial immunodiffusion or Mankin's method is also known as Mankin's immunodiffusion or single radial immunodiffusion assay. It is a single diffusion technique whereby a solution containing the antigen is placed in the wells. So antigens are placed in the wells and this is a slide where agar gels are poured. So here the agar gels are poured and then the antibodies are present throughout the slide, and in the wells the antigens are added. Okay, and the antigens will diffuse out and produce a precipitation ring outside this circle. So depending on the circle of the or ring size, depending on the ring size of the precipitating ring, we can say the action of antigen antibody reaction. So the diameter of the ring that precipitates around the well as a result of antigen antibody reaction correspond to amount of antigen in the solution. Okay, so move on to the next question, that is antigens used in well Felix reaction are OX2, OX5, OXA or OX2, OX5, OXC or OX2, OX15, OXY or OX2, OX19 or OXK. So the right answer is option D, that is OX2, OX19 and OXK. So a well Felix reaction is a type of agglutination test in which patient serum is tested for agglutinins to O antigen of certain non-motile Proteus and rickettsial strain, that is OX19, OX2 and OXK. Okay, and that are the strain of Proteus vulgaris. Okay, and the last question is C1 inhibitor deficiency causes systemic lupus erythematosus or severe meningococcal disease or recurrent pyogenic infections or hereditary angioneurotic edema, and the right answer is option D, that is hereditary angioneurotic edema. So C1 inhibitor deficiency causes this hereditary disease, and this acquired C1 esterase inhibitor efficiency, also referred to as acquired angioedema, is a rare medical condition that present as body swelling that can be life threatening and manifest due to another underlying medical condition. The acquired form of this disease can occur from a deficiency or abnormal function of the enzyme C1 esterase inhibitor. Okay, so this is the normal condition, and in the disease condition the woman is like this. Okay, so these are the all questions of this video. So thank you for watching this video, and we will see you on our next video, that is the part 10 of immunological MCQ. Okay, thank you for watching this video.